Good morning, students. So today's class we are going to study about the EEG signal characteristics. So EEG, it is nothing but electroencephalogram. It is one kind of bioelectric signals. So the EEG, we call it as brain waves, and it responds to electrical activity of the brain. The electrical activity of the brain can be studied by using this electroencephalogram. So, if you consider the brain, it consists of main parts like cerebrum, cerebellum, brainstem. The brainstem, which includes midbrain, pons, medulla, and reticular formation, and thalamus, which locates between the midbrain and the hemisphere. So, these are the main parts of the brain. And when you see the cerebrum, it is having two hemispheres. It is separated by the longitudinal fissure, so which is called as the fibers, connective band of fibers called corpus callosum. And then the outer surface of the cerebral hemisphere, it is known as cerebral cortex, so which is composed of neurons, which is gray matter, and it is separated by a regions called fissures or sulci. So these are the main parts of the brain. So in the brain, so at each and every outer surface of the location, that is skull, we will be placing the electrodes and then recording the EEG signal. And if you see the EEG signal, we can able to generate the cortical potentials. That means the amplitudes which is generated due to the excitatory and inhibitory postsynaptic potentials which is developed by the cell bodies and the dendrites of pyramidal neurons. So here the physiological control process or thought process or applying the stimulus which corresponds to the brain all are recorded in the scalp using the surface electrodes. So the electrodes will be placed over the scalp at different regions at different lobes, and then EEG signal is recorded. So you know there is a standard electrode arrangement placement arrangement is there. So we call it as 10 to 20 electrode placement arrangement. Using that, we place the electrodes in the different regions of the scalp and record the EEG signal. So the 10 to 20 electrode system, no, so we call it as starting from the nasion to the anion. So the distance between the in between distance we call it as 10%, 20% like that. So according to the International Federation of Society for Encephalography and the Clinical Neurophysiology, so they have recommended the 10 to 20 system of electrode placement for clinical EEG recording. So if you see the instrumentation settings, so we use low pass filter at 75 Hertz, and the paper will be recorded at 10 microvolts per centimeter, and the speed will be 30 per 8 for 10 to 20 minutes, and either 8 or 16 channels, simultaneous channels will be used to record this EEG. So we can able to record not only the sleep EEG, epileptic seizures, and other abnormal conditions related to the brain. We can able to record using this EEG signal. To record this EEG signal, so we can use needle electrodes or nasopharyngeal electrodes, electrocardiogram, which is placed over the exposed portion of the cortex and recording the signal, and intracerebral electrodes also used to record this EEG signal. So if you see the 10 to 20 system of electrode placement for this EEG recording, it starts from nasion and till inion. So if you see this FP represents the frontoparietal region, F represents the frontal region, C represents the central region, P represents the parietal region, O represents the occipital regions, and this side in ear lobe A1, A2 is nothing but it is a temporal lobe. So in all these locations, the active electrodes will be placed, and the reference electrodes will be placed on either side of the ear, which is called as temporal lobe. So mostly they'll use more number of electrodes which is placed over the scalp to record the EEG. So usually 19 active electrodes and 2 reference electrodes will be used, or even more than that also we are using this um, electrode arrangements, and more than 32 channels are there